All right, I just refreshed. Can people hear me, Mary? Oh, okay. Okay, good. All right. Let me just go back a few questions here because we jumped a little bit here. All right. Well, you guys can type faster than I can think. All right. So that's back to there. Yeah, Judith, there's not a lot of evidence for sex change in crabs. Some of the other crustacea, yes. Not a lot of evidence for it. But then again, not a lot of people keep enough crabs long enough and document individuals to really keep track of this. So it may be something that is commoner than we think. We just don't have the evidence for it, basically, which is interesting. Looking back at Stacy's comment, the female openings definitely come and go. Oh, interesting. That's something I have never heard of either. So you're basically saying an individual can lose openings between molts and then they come back again. That would be interesting as well. As you know, to change any of their external morphology, it has to be done at a molt. And so if you're going to lose your female openings and develop male openings, the only way you can do that is via a molt. And if you're going to grow a sexual tube, the only way you can do that is by molting. Yeah, if anyone's got any papers, I would love to receive them. I sent my email recently to somebody at the end of the talk, but let me just type it here now. And anyone who wants to send me anything or to discuss anything with me, more than happy to do so. Yeah, I would be interested to hear Mo's talk about this cetal pattern tracking. You guys need to be able to track individuals somehow. And I know that marking individuals can be difficult. Some research has done it by freeze burning, letters and numbers into crabs, and it lasts over multiple molts until it eventually disappears. That sounds kind of cruel. It's like branding. Of course, you can't do it with the shell because they swap them in and out. But the best bet would be if you have single individuals in tanks and not multiple individuals, and over time you can document changes in morphology, changes in sex, the coming and going of various external reproductive structures of the same individual. That would be very useful to see that. Yeah, intersex is basically hermaphrodite, although I did read that some crabs have been discovered that had an external intersex morphology, but internally they only had one of the reproductive systems. So it doesn't always follow that the internal reproductive systems develop concurrently with the external morphology. So there is some evidence that they don't have to match. Now, the functionality of intersex, that is interesting. Theoretically, if you have both internal and external reproductive structures for male and female, you should be able to use them both at the same time. We have no evidence that they can be simultaneously used, or even if they can do things like self-fertilization or anything like that. I know of no evidence of that whatsoever. That would be another interesting piece of information to get about individuals, but you'd have to know that they had internal reproductive organs that match the external, and of course the only way to do that pretty much is to kill them and cut them. So the only way you guys would collect that data would be if you had the death of individuals. You should be pickling them and saving them, and they could be investigated later. And I'm going to talk some more about that on Sunday, about data collection that could be useful for science. Yeah, I don't know anything about intersex frequency, Christine. there's 10 or a dozen papers on wild Cinnabita and probably nothing on captive ones. So a good point to start looking at this, asking your community to document frequency, and that would be a good way to start gathering some data. Yeah, as I said, Stacy, basically killing the crab or waiting until it died. If you're not up for being the grisly executioner, then you could definitely just wait until an animal passes, and if it still is in that condition, to keep it for posterity. I was talking to Mary about this the other day. The principal fluid of choice for keeping crustacean specimens is 70% ethyl alcohol, and rubbing alcohols. What is that? Not propyl. What am I thinking of? Propyl alcohol, I think it's called. Rubbing alcohol you get from CVS is 70%, and that would work as well. So if you all went out and bought yourself a liter of that, liter, that's my Australian coming out there, half gallon or less of some propyl alcohol, and keep it in the house, and some small glass containers with lids to keep them in, you should be able to keep all of your organisms that pass, and they may at some point be useful to somebody. I see a question or a statement here from Robin. Maybe they lose their gonopores when they aren't healthy enough to produce eggs. That is an interesting idea. From an evolutionary perspective, when you think about it, why produce a structure that you're not going to use? And if you're not healthy enough to reproduce, then they should not be wasting energy on developing ovaries and eggs and reproductive structures. But when you think about it, a gonopore is such a small investment from an energy perspective. I can well imagine a sexual tube being a much greater energy investment, but the female gonopore disappearing seems like a very small sacrifice to make, when I think other major sacrifices will probably be on top of the list for that. I'm just scrolling through these questions now. Oh, absolutely, Stacy. Somebody asking about captive bred babies to track through life. One thing I forgot to mention when I was doing the study on the Cinnabita pilatus from the Smithsonian is we had no juvenile individuals in the collection, because, of course, there would be a collection bias on larger crabs when whoever went out and collected them. They were collected over a 120-year period at the Smithsonian. So the first people who went out to collect them would, of course, been collecting the biggest and most obvious individuals. And we had some individuals that were in shells nearly as big as my fist. But the smallest individuals were still mature individuals, the males. And so they all had sexual tubes. And so it'd be really interesting to see when a sexual tube develops in a growing hermit crab from when it's a juvenile to a male. Now, a juvenile, by definition in biology, is you are non-reproductive. So a juvenile should not have a sexual tube, or should not have a functional one or a functional reproductive system. And then as soon as it does, no matter what size it is, it then becomes no longer a juvenile and it's an adult. Even if it is a tiny size and it has a sexual tube and it has gonads or ovaries, it is an adult. So it'd be really interesting to track the development of individual male and females through the juvenile phase to see when these structures start to grow. And if this is a sexually selected structure, then any male hermit crab should prioritize its development as soon as it's sexually mature, because it obviously has a functional role. So they should start to invest energy in growing a sexual tube as soon as they're capable of reproducing, no matter what size they are. And so you should see this almost instantaneous arrival of a sexual tube, and then this disproportionate growth should occur. And you could easily track that once again through individuals of a known cohort over time. Once again, you either wait for them to die, or if you're a grizzly, you can be an executioner and you could collect them, and you would be able to find out when exactly that transition occurs from juvenile to adult. So the fact that you guys are now breeding them is a great opportunity for some research. Although I well recognize that these are treasured individuals, and I need to lose my research perspective a little bit about them as subject animals, and remember that you guys are connected to these, and I don't want you just going out and performing mass infanticide for research. So I'll keep that in mind. I see a comment here from Dan Comos about interbreeding. This is another interesting thing that Mary mentioned to me the other day about everybody in your community saying that there was a lot of examples of hybrids and hybridization. I can't think of any literature that's been published about known hybrids in the field, but remember, a lot of these species are very disparately spread around the world and disconnected from each other. And so only by bringing them into captivity and putting them into the same environments are you putting them into positions where they could possibly hybridize. For example, a Cinnabita, Clypeatus from the Caribbean is not ever naturally going to meet, a Rugosus from the Pacific or whatever, or a Pilatus from the Indo-West Pacific. So natural hybridization is unlikely to occur because of their disparate biogeography. But when you start bringing them into captivity, just like lions and tigers and ligers and tyons and all these sorts of things, you will get closely related species that can possibly hybridize. And that would be an interesting set of scientific results as well. You could definitely tell a hybrid simply by sequencing them, doing their molecular genetics. Each species would have a unique sequence, and hybrids should show a mixture of the two. And so keeping young individuals that die that you think are hybrids and pickling them in alcohol, that is also the preservative of choice for molecular genetics, 70% alcohol. So at a later point, somebody could do some extensive sequencing of all of the species and also the hybrids to see if we can show that they are hybrids. And if it's not unknown, even in nature, that closely related species can still hybridize. It all depends on how long they've been separated from each other. From an evolutionary perspective, it's reproductive isolation that creates species. When you going back to your high school biology classes and lectures on speciation, you need reproductive isolation to develop speciation in general. That's the commonest form. And so it is the isolation of different species over time that generally drives them to be different. And so some of you may be bringing these species back together to hybridize ones that may have been separated at some point in their evolutionary past, which is kind of cool. So another thing that you guys can contribute to. Oh, that's good to know, Mo, about tracking individuals with CT patterns. That's really cool. Yeah, all you have to do is produce a series of images over time showing the same individual with the same patterns, and then prove to me that it changes sex. I'll totally believe you. Absolutely. I see Mary's comment there about more likely for sex change in captive hermits. I mean, that goes, there's a lot of stories about captive animals doing that because of stress or reduced breeding opportunities, etc. You all remember the dinosaurs in Jurassic Park, right? So yes, if they are capable of sex change, then you need some sort of environmental cue to make them change sex. And captive individuals may be getting that cue more frequently than wild ones are. So yeah, that's probably true. And once again, you guys have that data. You know who's in your tanks when, how long they've been there, what stage of health they're at, to know whether particular individuals have changed sex under particular conditions. So you need to document all that. You need to start to write all this down, keep it in notebooks, type it up, and store it all. And eventually somebody someday is going to ask you for that data. So looking at a question here from Christine Sandberg, what percentage of male versus female in the Smithsonian study? In general, Christine, I'm really not interested in females. I've been a male reproductive biologist since my PhD days, and I am interested in general in reproduction, and I have done a little bit of work on females, but generally all my work on hermit crabs has been male internal and external reproductive structure, and I've done very little on eggs. So we weren't actually interested in the females. My student went there, looked at every one of those hundreds of hermit crabs that were in the jars there, and simply selected out the males, measured their carapace length, measured their sexual tubes, and put them back. So I don't actually know the male-female ratio for that. It should be approximately equal, although in some species, in some environments, there can be a lot less males if there is competition for females and the same males are breeding repeatedly with other females. Oh, absolutely, Stacey. If anybody is raising broods and they're dying, you should be keeping them. You should also be keeping, if possible, molts. Now, I know they're very secretive about molting, and I know they disappear for days or hours and then appear with their new clothes on, and probably eat their old molts for the minerals. But if you can get hold of molts, that may be a really definitive way to tell an individual has changed sex if you can keep all their molts over time. That would mean stealing a molt off somebody after they have molted before they can eat it. And you should be able to track that those external cues of gonopores and sexual tubes and pleopods, etc., which should tell you if they have changed sex or not. But I know it's difficult to you suddenly realize a crab's gone and a day later it's molted. So if you can, you should be keeping your molts as well and preserving them the same way. You can dry them out, but it would be better in 70% alcohol. Dead hermits in the mail, Christine. I've been getting dead hermits in the mail for years. All of my PhD research, people collected hermit crabs from me all over the world and posted them to me. Back in the days when posting anything to the mail was pretty much possible, I got hermit crabs from Numia, New Caledonia, where else? Indonesia, the Mediterranean, Brazil. I was getting dead hermit crabs in the mail all the time. And if I wasn't getting dead hermit crabs, I was getting gonads in the mail. People would put hermit crab testes in a jar and send them to me when I was doing comparative sperm analysis for my PhD. So dead things in the mail is no problem for me at all. So there you go. Oh, I see questions come to the top here from Stacey about pheromones. There's a couple of great papers out there about the smelling ability of land hermit crabs. They have an amazing ability to detect airborne odors and attracted to the bodies of their dead conspecifics for shell acquisition, and also to food types. They can smell particular food types, dead animals and fruits and things. So they have a great ability to pick up scents. And you know those two antennae they have in front of their eyes that they're constantly flicking, they have little structures called astrotasks on them, which are sampling molecules in the air. And basically, they think they flick them to reset them. So air touches them, molecules land on them, they're tasting them, and then they flick them to clean them, and then they're tasting again after that. That's the current theory for why they're constantly flicking their antennae. So it wouldn't surprise me if they're producing all sorts of chemical cues for all sorts of activities, including when they're fighting for shells, when they're trying to find out who's a male and who's a female, when they're actually engaged in sex. There's probably all sorts of chemical signaling going on there. Marine crustaceans do it. It's well known. Lobsters, male lobsters send urine streams across at other male lobsters to intimidate them, basically a pissing match. And it's very likely that terrestrial animals are doing it as well. It's just that it's harder to do that in aerosol form than it is in a liquid form. And so it's very likely that they're doing it, but they may have to do it over shorter distances because the aerosolized chemicals could very easily be dissipated, more so than they can be in water. Oh, these things are coming fast and furious here. How much of a role, here's a question from Darcy, how much of a role do you think the environment plays in reproduction? All I can probably think is that if a crab's not healthy, it's probably not thinking about sex. So I well imagine that environment has to be providing a healthy resources, and you have to have a physiologically healthy crab. But as I said, I only deal with dead crabs, and I have never kept crabs to reproduce. And so you guys are in a much better position to answer that question than I do. When I was a young boy, I used to breed birds, finches, and we used to put ground black pepper in their seed just before breeding season because apparently it made them breed better. So you guys probably have all sorts of secrets about what to feed your crabs to make them breed. But that's a question for you guys to answer. I don't have an answer to that whatsoever. Question coming up again from Andrew. Is it possible for them to crossbreed? As I said, if you've got them in captivity, it is very likely. If they are from the same part of the world and recently speciated, they may still be able to breed effectively, and they may still be able to produce offspring. Whether they're fertile and viable depends on their period of separation as a species. That's the general rule for hybridization and speciation. So things like Pilatus and Rugosus, if I remember, are both from the Indo-West Pacific. So any of these species that even, especially ones that coexist in particular regions, the southern islands of Japan, like Okinawa and places like that, Guam, have multiple species on them, I believe. And the Australian coastline has two, Variabilis and Pilatus. So anywhere where they're cohabiting, that might be possible. But remember, evolution is going to give them mechanisms to prevent that because it's not very efficient to constantly hybridize. So there will be all sorts of naturally selected mechanisms. It could be pheromones, it could be behavior, it could be sound, it could be timing, nighttime mating versus daytime, seasonal changes that will be in place to prevent natural crossbreeding. And you may be able to circumvent all of them in captivity. Like I said, lions and tigers don't normally live in the same place. If you put them together in the wild, they're likely to kill each other, but you put two of them in a comfy Las Vegas hotel room long enough, and you might end up with ligers and tyons. So captivity might be the answer to that. A question from Mo has made it to the top here again. Information or thoughts on why pilatus sexual tubes in the size and shape they are. Wow. There's about 12 or 14 species of Cinnabita, and only a couple of them have those long sexual tubes. Most of the species have symmetrical, large, flat, square coxal segments that have bumps on the end, but not those tubes. So, no, I don't have an answer to that. If we can find out what the pilatus are using it for, as in how is it being used during reproduction, we may be able to answer why they have it. But, my answer would be if they have it and they need it, why don't the other species have it too? And that is an answer I do not have. That is the million-dollar question. And the same thing for those aquatic hermit crabs. All of those members of the family Paguridae, about 48% of all the species in the family Paguridae have been documented with sexual tube, but the other 52% don't, and they breed perfectly fine. And they're all sorts of sizes, and they live in the same environments, and sex for them occurs without them. So why do some species have them and some don't? That is the million-dollar question. That's what I'm really interested in from an evolutionary perspective. Let me look at some of these questions here. Oh, Mary's put one at the top there. Do you have any thoughts as to how often a male can mate? They have prodigious amounts of sperm and spermatophores inside them. As I said, a single large spermatophore of a pilatus, for example, which has got a fairly probably has less than 10,000 sperm cells in it, and they probably pass hundreds of spermatophores over to the female. So let's say there's 1,000 sperm in let's say there's 10,000 sperm in one spermatophore, right? And they pass 100 of them over, right? So does that make 1 million sperm cells? You guys who are good at math. And if the female's only got 400 eggs or 300 eggs to produce, then there are more than enough sperm cells to go around. But once he's passed them to one female, he can't pass them to somebody else. But when I've opened up male hermit crabs, they are literally filled with tens of thousands of spermatophores. And so, they have great potential to fertilize a lot. It would just be a case of time and availability of females. That would be the limiting factor, I would suspect. And they probably have great potential to recover. There's all sorts of studies out there about sperm limitation in males. Because males are often competitive and certain males get more matings than others, there's the potential that those males will actually deplete themselves because they're out competing smaller males to have access to females. But then when they get the females, if they mate with too many of them, they can't perform when they have that opportunity. There's often little males hanging around the outside to take advantage of that. They're often referred to as sneaky copulators, and they will whip in when the big male has finished and try to deposit on the female when basically she's not looking. But if the male is unable to produce because he's just mated with too many females in too quick a time, then those other males may take advantage of that. As far as I know, no scientific evidence of how to quantify that sperm production and sperm depletion in them. It's a really important factor if you're going to try and breed them, because you want to know if you should be using a single good breeder all the time or whether you should be resting them like a good thoroughbred racehorse or not. But not much information about that. And Abby, as I said before, I haven't really been paying a lot of attention to the female reproductive biology of Cinnabita. There might be a few papers out there that quote egg numbers on the species that are there. I have a couple of papers that talk about reproduction in Cinnabita. They're a little old now. There was a guy named Nakasone, a Japanese guy, Nakasone, I think. I think he produced 2 papers looking at reproduction. I can't remember if he quoted female egg numbers. I have those papers somewhere. If you wanted to send me an email, I could look them up and see what's in there. Or you might look it up yourself, see if you can find papers by Nakasone, Nakasone. Do it on Google Scholar and put Cinnabita after it, and you're bound to pull up, here's 2 or 3 papers, and you should be able to find them. Interesting. Somebody had 2 different species exhibiting mating behavior. Yeah, I mean, like I said, put them in a small tank, play nice music. You never know what they're going to get up to. Whether they can successfully mate, you have to prove that with your babies you're producing and whether they are hybrids or not. I presume you can tell hybrids by a mixture of external morphology, maybe like colors. I know you know very well that these have very unique color patterns as adults. And so you probably can see hybrids because they have a mixture of those external color patterns. You might also think about looking at males to see if their sexual tubes are a hybridization of 2 different ones. So, for example, a Clypeatus and Pilatus, as you just saw in my talk, have very different sexual tubes. So if you raise young male hybrids, what is it going to look like? It should have a hybridized sexual tube as well. Once again, you guys have the answer. Get to it. So how are we doing here? Have we run out of questions yet? Just scrolling down here now. Yeah, we have a mission now, guys. Do you ever? Yes, I want you to prove sex change. I'll be happy to help. Mary, that's a great quote. Nature always finds a way. Is it Goldblum, wasn't it? That was that. He was the mathematician. Yeah. Yeah. Interesting, Moa. You don't believe in hybrids. Well, like I said, it depends on where you're getting them from. So interesting. Yeah, but as I said, you said, where you've got different species living in the same place, evolution and natural selection would have deliberately given them mechanisms to prevent hybridization in most circumstances. So I think you'd only get hybridization in captivity because you have a relaxation of those mechanisms. I think that would be my answer to that. But yes, in nature, natural hybridization is unlikely to occur in disparate populations, and it's unlikely to occur, except under extraordinary circumstances, in cohabiting populations and species as well. So, but it is interesting. If you can get hybrids in captivity, that's really interesting. It would be interesting for your community and for the possibility of selling hybrids. You'd have to find out if they were fertile or not, of course. But you could do some very interesting hybridization, and you could make, just like dog breeds, you could make things that people were more likely to be interested in or not. And as I said earlier, you may be able to circumvent the wild caught industry by providing unique stuff that they can't get. Question from Eric statement from Eric about 10 minutes ago. I thought they needed their old exoskeleton for regeneration. Most likely there's minerals in there that they need to recover. As you know, the crustacean cuticle is a combination of calcium carbonate and minerals and protein. It's a complex. And they're shedding off a lot of the minerals in there. And if those minerals are not available readily in their environment, it may take them quite a while to recover those minerals naturally from their diet. Then they probably need to eat their old exoskeleton, or at least the parts that are easily edible, to recover some of those minerals. And remember, when they molt, they're soft, including their mouth parts. So it's limited what they can eat until they harden. So as soon as their mouth parts start to harden, then they can chew on regular things, and then they can start to get back the minerals that could be in their exoskeleton. I suspect that immediately they cannot eat their own exoskeleton because they can't operate their jaws and they can't chew on hard things. An exoskeleton works differently. It's to an endoskeleton. You have an endoskeleton. You have bones on the inside, and all your muscles have to attach to it. And that's the only way muscles can work, to be attached to something hard that doesn't move when you contract. An exoskeleton works the same way, but the muscles attach to the outside, and you harden it, and the muscles act like bones, and your muscles act against them. If you make your exoskeleton soft, then your muscles can't operate very efficiently against them. So newly molted crabs can't move very much. They're very vulnerable, which is why they hide and dig holes and crawl back into caves, etc. And so I well imagine they also can't eat effectively until they start to harden and mineralize their bodies again. So they probably need the old exoskeleton for lost minerals. Now, if you provide an environment with lots of those minerals, they may be less likely to eat their exoskeleton, and you might not feel so bad about taking it off them if you can find it. Taking a fresh exo from a newly molted crab could be dangerous. As I said, you might want to leave it a little while until it's at least capable of movement. Oh, Stacy, good. You read those Nakasone papers? Very good. I haven't delved into the Cinnabita literature in a long time. I'm actually moving my office right now. We're moving into a new science building at school, and I'm currently packing up and throwing away. And I actually found my Cinnabita file just the other week. It's got about 20 or 30 papers in there, ecology, behavior, etc. And so I saw them again. I packed them into a box, and they're going to be moved in the next couple of weeks. But I haven't delved into Cinnabita stuff in quite a few years. All the hermit crabs, something from Abby, with all the hermit crabs dying in plastic trash. Is this in nature, I guess? We're talking about plastics in the ocean, washing up on beaches, and them finding themselves bits of plastic to crawl into. Like any other creature, I think that plastic is detrimental to them, both chemically and physically. So, yeah. I know, for example, when I was an undergrad student, I went to the Great Barrier Reef in Australia. I went to Heron Island, which is a wonderful place. It's half national park, half resort. And then my university, the University of Queensland, has a research station there. And I did several courses there as an undergraduate. And wandering around that island at night let me go back, let me date myself now. We're talking about 83 through 86, I guess. 83 through 85 or 86, there were a very small population of Cinnabita pilatus living on Heron Island, which is a very small tropical cay. And there were so few of them, they had each individual labeled and numbered, essentially, and the national parks people were doing a study of them. By the time I came back as a graduate student in the early 90s, as I was a postdoc actually, they were gone. There was no record of any pilatus being left on the island. So there was probably only tens of individuals when I was there in the 80s, and a decade later, they were all gone. So, and I was just back with my family 2 years ago on a holiday there, And I talked to the people at the research station and at the resort there. I didn't get to see any national parks people, and nobody there can remember ever those crabs being there. So there's no one old enough there on the island who remembers the population being there. So these populations are vulnerable, especially on small islands and small populations like that. You can well imagine that pollution could get rid of them on some of these isolated islands. Oh, Dan, that's a really good question there. Do we know of an average size or age when crabs are mature enough to mate? So as I said before, as soon as they molt from a juvenile to an adult, and that could be tiny, they could be the size of a pea or smaller. As soon as they've got the equipment, they're capable of mating. Now, they're only capable of mating with similar-sized females at that age because they're most likely not going to be competitive with larger males for access to females. So it all depends on the mating behavior and strategy. If they get together in large groups to mate and the males are competitive and push each other around for access to females, then those little guys are not going to get access to females. If, for example, you exploit a population and take all the big ones out of it for, say, the pet trade, and all you're left with is smaller individuals, then those smaller individuals are then competitive for mating, and the larger ones of those would be the ones that would be most successful. But literally, as soon as they have their equipment, they're capable of reproducing. Whether the reproductive behavior allows them to, gives them access to females, is a different story, and also might depend on population size and sex ratios and things like that. Wow, some of us have witnessed males being extremely involved in spawning process, holding the female underwater as she releases eggs. Well, I hope that's what he was doing to her. It sounds like a homicide to me. I would imagine that males should not be involved in the spawning process whatsoever. They shouldn't care about their offspring, and they shouldn't care about any female that they mated with, only the fact that she's not available to mate with anymore. That surprises me, although once again, in a captive environment with a limited number of individuals, you may see very different behavior in that regard. A male guarding her. Okay, mate guarding. That's an interesting thing, Stacy. That's very common in a lot of crustaceans, especially when mating involves a soft-shelled female. This is not the case for Cinnabita, as far as I'm aware. The female does not have to be an intermolt and soft for sperm deposition to occur. I could well imagine that it would be beneficial for the female to be hard, for the male to have somewhere to put his sperm mass. But crustaceans that can only mate when females are soft, the males often will stand over the female and mate guard her until she's hardened, because he is preserving his paternity over somebody else's. Because if he walks away from her while she's soft, she's vulnerable to mating by another male or being eaten by another crab or some other predator, where he's then lost his paternity as well. So I don't know of any mate guarding. Once again, you guys have seen this. You've got the documentation of it. I haven't. If mate guarding is occurring, that's really interesting. And I don't know if that's a natural behavior or it's one that comes out of captive environments and limited population sizes, etc. I thank you for pinning stuff to the top. That is great, because I've just been scrolling down and finding stuff. I think Robin already answered about the sense of smell. There's a couple of really good papers out there, I think from a guy named Greenaway, an Australian guy, and some others, on their ability to discern aerosolized smells, and they have a very good sense of smell. So that wouldn't surprise me if they can smell any food type over a distance and be able to discern different types of food. Oh, I love this, Stacy. You've got some mating behavior stuff there. That is very cool. Yeah, you should be videoing all this. Wonderful. I don't think anyone's done this in the wild at all. I suspect all of this is happening at night, possibly undercover, in holes, under leaf litter, etc. But you guys have them behind glass and available. I could well imagine that when a male finds a receptive female, he's going to try everything he can to be the one to access her. And so it's very likely that they would be following females around and enticing them. They can't forcibly mate with them because the female, of course, can pop back in her shell, seal herself in, and she won't allow the male to access her. She has to come out far enough for him to deposit onto her ventral surface. So she has to be a cooperative female, and he has to be a cooperative male. As far as I'm aware, you may have other stories that go against that. Oh, that's interesting, Moa. So you've got different species in the same tank, and one of the females is ready to mate. Only males of the same species are interested. Yeah, that wouldn't surprise me at all. They probably are releasing species-specific pheromones. We know insects do it, and male moths can track a female down over miles based on the pheromones that get released. So within an enclosed tank, that doesn't surprise me at all. Although, as I said earlier, you may get exceptions to this in captivity over time. Here's something from Brooke. Are there any other studies going on at the Smithsonian about hermit crabs? Well, as you know, the Smithsonian's shut down right now. I haven't been able to go there since the beginning of the year. My colleague, Raphael Lemaitre, who was one of the world's leading hermit crab taxonomists, just recently retired back in February, just as COVID was hitting. So they now have no hermit crab people employed at the Smithsonian. So I can well imagine that my research there is going to really start to wind down. Although he and I have a plan to actually publish a major review on sexual tubes. We wrote it 10 years ago, shelved it, and then both of us got so busy we never got back to it. But he said it was going to be the last thing he wanted to do when he retired. And so he and I might be working on that. But there's no other people doing hermit crab stuff. There's a guy in up the northeast somewhere named Mark Laidre, Laidre. He's very interested in land hermit crabs, their sexual tubes, their sexual behavior. Here's a really interesting paper about penis size, which is what he calls a sexual tube, and what the influence of that is on shell quality or house quality in these. He's written a couple of papers on that. He has still got graduate students doing work on that. And then, as far as I know, there's a couple of Japanese working on the Japanese species in southern Japan, Okinawas and places like that. But there are no active hermit crab people except myself, and I'm an associate at the Smithsonian. I'm not an employee, research associate. So nothing else going on there as far as I know, and very likely to wind down now. Okay. Oh, Stephanie, you wouldn't believe how many times I thought about doing hermit crabs myself, and the thought of the time commitment and the equipment commitment and the fact that I've got my fingers in so many other pies at the same time, I blanched at the thought. It would be so much fun to keep them and to do this. I imagine it'll be a full-time job. I have several active research areas going on at the same time, and unless I had some really devoted students, which I haven't had at AU so far, no, I haven't, not in a long time. It would be great. Do you know something I thought about for you guys as well? Do you know that you can get glass shells now? Of course, they make them. You can buy them and get them. That would be great to be able to see what males and females are doing inside their shells. So I know it's probably an anathema to most of you to put artificial shells anywhere near your crabs, but from a research perspective, being able to look in and see is very interesting and might enable some videoing to go on there. I always thought about keeping a population of them and giving them glass shells so I could observe them. But I'll leave that up to you guys now. I want you to collect all that data. Oh, I'll have to come back and look at that cooperative spawning later. I haven't got time to look at it right now, but Mary, I will come back and look at that. The chat's being recorded as well, correct? I should be able to come back to this later and do that. Oh, I see a comment from Judith there. Yes, you're right, Judith. They do have organs that they store a lot of those minerals in, so they don't waste it. They pull as much as they can out of their old shell. You're absolutely right, before they dump it. But there's still some in there that they can recover later. They pull things back into their hepatopancreas, which is basically their liver, and other places in their body to retrieve it later. Oh, you've got several hundred papers, Stacy? Oh my gosh. Do you have a bibliography of that? If you do, if you could share it with me, that'd be great. I don't have several hundred. I probably have about 30 or so papers, but I've only been collecting from the scientific literature and only the stuff associated with reproduction. So there's larval papers out there, and there's ecology, and there's probably some behavioral stuff too that I haven't been interested in if it wasn't associated with reproduction. But if you've got a bibliography of that, I would love to see it. Moa brings up a good point. If you're really squeamish about taking the overcoat away from your crab before they're finished with it, photographing it, a good photograph of the ventral area before they've eaten it might be a good substitute for it. Oh, there's some really good information here. I'm just reading through some of these now. Oh, that's interesting. Who just wrote that? Brooke. If you only have one male in a tank, that's interesting. They may not exhibit guarding behavior if they know they're the sole reproducers. So that would be interesting data to collect as well. If you guys have multiple males or single males, does it really make a difference whether they guard or not? All right, scrolling down here. Everyone's looking at this video now. I'll have to check it out later. Oh, Jennifer Nemec had an interesting comment here about sex change and whether they're more likely to be males or females at a smaller size, which is quite interesting. Individuals that are hermaphrodite or do sex change, the norm is for them to be males first and females second, because the investment in sperm from an energy perspective, the percent of the body mass, is a lot smaller than the investment in eggs. And so the bigger you get, it pays you to invest more of your body mass in eggs than it does sperm. Where that falls apart is where access to females is determined by body size. So if big males get more breeding opportunities than smaller males, then you may actually have a switch in that norm, and that it might pay them to be female when they're younger, and then when they get big enough, they can be competitive and switch to male. So once again, no data on that, just hints from other crustaceans and other invertebrates. There's all sorts of interesting analogs in nature. I think Mary brought it up in a conversation we had weeks ago about wrasse, and everyone knows the fish, the wrasse, in tropical environments, there's often a dominant male, a dominant breeding female, and a whole lot of subdominant females that don't breed. And if you remove the male, the dominant female turns into the male. All of the subdominant females fight it out until one becomes the breeding female and becomes the dominant female, and then she becomes the breeder only. That's a model that could very well work in some environments, and I don't know of any crustacean analogs of that, but there's all sorts of interesting models like that, and it can work in different ways. Oh, interesting about the glass shells, Christine and Stacy. Yeah, I really would only be thinking about it from a research perspective. So, as I said, yeah, you guys have the knowledge and the experience whether that's going to work or not. Stephanie, a comment from you. I wonder if the sexual organ size may have to do with the shell type the different species prefers. Well, in some cases, beggars can't be choosers. In many populations, shells are a limiting resource. As you well know, there's lots of papers about land hermit crabs in some places, putting themselves in man-made objects because there's not enough shells around. So shells, even with aquatic species, can often be limiting. If you had unlimited shell access, then it's probably not going to be a big deal. But if you really, shells are a rare resource and you have to keep what you have, then it may actually have an impact on what you would prefer and what you're stuck with, and whether you can actually molt and grow effectively or not. So that's interesting. I don't know if there's any data out there on that either. Mary's just popped one up to the top here. Is there a correlation between sperm size and egg size? I wish I knew that. I don't actually know egg size dimensions. That's something you guys can collect data on. I do have the sperm size for nearly all the species of Cinnabita, well, at least 5 or 6 of them. I did that for my PhD thesis a million years ago. So I can probably provide sperm size data, and you guys can provide the egg size data if it's not already published out there to look at that. There's not a great difference between the sperm sizes amongst the Cinnabita. It's a small difference between different species of Cinnabita. The coconut crab, Bergus latro, did have a proportionally larger sperm than the other Cinnabita species did, but we're talking about maybe just a 20% difference in size. So you, Mary, you said here that you saw that the sperm of pilatus was one of the largest sperm. You didn't actually see, you only saw the sperm of Bergus latro, the coconut crab. I didn't show you any sperm of Cinnabita. What you were looking at was the spermatophore. That was that picture that had 5 or 6 of them on there. And Bergus latro had the biggest spermatophore. I think pilatus had the next biggest, and then they were going down size after that. So that's not sperm size, that's spermatophore size. I can go back and look at my thesis and find out the lengths of all the different species of Cinnabita, but they weren't very different from each other. So there wasn't a great difference in sperm size. But, that's an interesting thing. You guys could gather that egg data very easily, pickle some eggs and measure them, or even measure them before they're pickled, because there will be a slight shrinkage. 10 to 15% shrinkage in alcohol when you put things in. Oh, the connection between Stacy here, the connection between egg size and larval development, that's really interesting because, yes, if you're going to abbreviate larval development, you need to provide more resources for your offspring. So that's probably a connection there. Although I do know that in some crustaceans, abbreviated larval development can often be in response to difficult environmental conditions. So while they're floating around in the plankton, which what they would normally do, if conditions aren't right, they will sometimes either do 2 things: abbreviate development to get out of it really quickly and drop down as a juvenile, or they will add additional steps to hang in the plankton longer to try and get through the event and then drop out as a juvenile. So the length and number of larval stages, instars, may vary depending on environmental conditions. I actually read a paper recently on that, and so that would be interesting to look at. And I don't know how that's going to relate to you guys raising them in captivity, because as good as you are at it, you don't know what sort of stress you're putting on your young larvae. That could be influencing the number of stages and the number of molts they go through, etc. Oh, interesting. Yeah, I think I heard of the Hamasaki paper, Stacy. So, yeah, there's a few papers on larval development in Cinnabita, and I'm just trying to think. There's also one on the transition from larvae to shells, and the twisting of the abdomen and things like that. I'm just trying to think of the guy's name who did that. But anyway, yeah, I can remember a few of those papers I have in my files somewhere. Oh, the correct environmental conditions for raising larvae. I mean, really, this is Mary's area. She's done it. But what you might think about is where your crab naturally lives, what the annual variation in salinity and temperature is, and if they have a breeding season, which you may be able to find out from the literature, what those conditions are during that season. So if you're trying to mimic natural breeding conditions, for example, you want to find out, where does Clypeatus live? When does it breed? What are the average salinity and temperatures, ocean temperatures during breeding season? And that goes for any of the Indo-West Pacific crabs as well. I would imagine that trying to meet that condition is going to maximize production. But like I said, Mary's done it. She's the expert on that. You need to talk to her. It may be very different for captive animals. Producing natural conditions may be detrimental to them, and they may prefer something different in captivity. You guys will have to find that out by trial and error, I suspect. And remember, you've done really well to raise these. No one else has ever done it. And remember that in nature, if a female produces, 2,000 eggs, there could be a less than 1% success rate to adulthood. So there's a great loss naturally. If you lose a lot of your larvae, that is natural, and you're likely to have a greater success rate than any individual female in the wild is. So keep that in mind too. Yeah, Stacey, it's very small. It could go up and down a couple of percent, but it really is small. It's the same sort of thing for sea turtles and stuff like that. It's natural selection, and if every sea turtle that ever gets born survives, then within a few years we'll be up to our necks in sea turtles. So there can't be 100% survival. There has to be attrition. They feed food webs, et cetera, et cetera. So it has to be small. Otherwise, we wouldn't have the biodiversity we have on the planet. We have limited resources. So something from Abby, can a hermit crab hatch with birth defects that will stay with it through its lifetime? I have no idea. Mary's going to have the data set to look at that, I suspect. Oh, interesting. I think Mary mentioned something about a one-eyed crab that didn't regenerate a second eye. That would be a genetic defect inside for sure, because usually most limbs can be regenerated when they're lost. But there might be some limitations on whether that loss occurs as a larval stage or juvenile. But it could be very much that is a genetic defect in that individual. So, Mary, don't forget to tell these people if they want to come and talk about this sort of stuff. I guess this is the crowd for Sunday, or am I making a mistake here? And it's a limited gathering on Sunday. Oh, very cool. Oh, Stacey, it was a lot of fun. I don't know if Mary told you guys, it's quite a contorted path that got us together with several people approaching me to talk to me about hermit crabs, and they told me about Mary, and I didn't even know about it. So we sort of got together through several other people, journalists, in fact, and I have so much respect for what Mary has done because no one else has done it, and I think it's fabulous that your community is so supportive of you, and I think you guys will go a long way. Right. Good idea, Darcy. I'm really concerned about natural populations. When I see populations disappear from remote, coral keys on the Great Barrier Reef, which is really not very impacted by humans, I worry about these places like the Caribbean and Indo-West Pacific, Indonesia, and places like that, where the impact on wild collection must be phenomenal and probably is going totally unrecorded. And it'll just come to a point where those species will no longer be in the pet trade because they have decimated the populations. I had questions initially a few weeks ago about is this a regulated industry, et cetera, and I think if you can circumvent the industry, then you're going a long way to saving a lot of these species in the wild. So you guys going to let me go now so I can go and help my wife cook dinner? Or are you going to keep me captive for a little while longer? Oh, is this this is a video. Oh, thanks, Mary. Look at
Sorry. Oh, did you want to see them? I was going to say goodbye, but yeah, I'll give you a look.
So is this all your babies?
Yeah, well, it's some of them. I just had a live baby cam going for everyone that wanted to see them in action. So they're just sitting here with me on my computer desk, but you can see the various size differences, and they're pretty active.
Yeah, they're very cool.
But
this is Mary who bred. This is my wife, Karen. This is Mary who bred those.
I just wanted to thank you so much. I also wanted to direct you to talk we have on Sunday, which is an amazing one by Tammy White, who is in Florida, and she actually bred Cinnabita lila and Cinnabita clypeatus in 2009 and was very modest about it, and none of us knew. And she has the most amazing footage in this talk of it's under a microscope, and you can see like a stage 5 Zoe transitioning to a megalopa in real time as he's breaking out. It's just, it blew me away. It's incredible. You definitely want to try and catch that on Sunday.
Yeah. What was her name again?
Tammy White. She runs
just WHITE.
WEICK, but you won't find anything online. We didn't. We all thought we knew what we knew. Okay. She hasn't done. She hasn't advertised it at all. She might be in here. If you're in here, Tammy, hi. But yeah, it's just really incredible footage that I asked her to give a talk. I had no idea what she was going to talk about. When I saw her talk, it blew the top of my head off, and I didn't even know she had done that. Carol Ann Orms apparently knew, and I missed that. But so we were revising hermit crab history. She got 24 C. lila to land and 27 C. clypeatus. So, and she thought they were Procura because this was 2009 before they even identified the lilas. So
yeah. Oh, I see Tammy there now. All right. Well, apologies, Tammy. People only told me about Mary. So it's not who does it; it's who gets mentioned by the press. That's all. So yeah. No, it's a lot of fun to be engaged with you guys, and I'd be happy to continue from this point onwards. And I look forward to chatting some more on Sunday. And I will drop in and see Tammy's talk. I'll go and check what time that is. And so that'll be fun.
Okay. Thank you so much. This was incredible. What a great addition to the conference. We are just honored and thrilled.
Well, it's fun. Somebody mentioned something about crazy crabbers, and I've been a crazy crabber since I was 20. So just on a different scale, that's all. So yeah. Thank you, everybody, and keep in touch, anyone, and especially you guys. All right.
Thank you.
I'm gonna go. I gotta help the wife cook.
Go enjoy dinner.
All right. See ya. Now, how do I get out of here? Leave. There we go.
Up in the top.
I see leave. I'm leaving.
It won't let me leave. I'm trying to leave. I'll close this out.